Welcome, and thank you for joining us today for the Harnessing the Power of the WHI Life and Longevity After Cancer Study to Better Understand Cancer and Aging webinar. My name is Jennifer Schaeffer, and I will be your WebEx technical support host. Before we dive into today's webinar, I wanted to provide a quick information on the webinar platform. All lines have been muted upon entry and will remain muted for the duration of the meeting. Please submit your questions throughout the presentation using the chat or Q&A panel and select all panelists from the dropdown. A moderator will ask your questions or comments on your behalf at the end of the presentation. If you need technical assistance, please feel free to contact me, the host, via the chat box, and I'd be happy to assist. Please note that closed captioning is available in the link in the chat box and will be posted multiple times throughout the webinar. This webinar is also being recorded and will be posted online at a later date. And with that, I'll turn it over to Jen Guida to introduce our webinar series and our speaker.
Thanks, Jennifer. Good afternoon, everyone. Welcome to the National Cancer Institute's Perspectives on Cancer and Aging webinar series. We host this webinar to share our broad interest in cancer and aging research and to honor Arthi Hurria's pioneering and impactful leadership of geriatric oncology, as well as to highlight her legacy as an impassioned clinician, researcher, and mentor. As such, each webinar in this series showcases complementary research or clinical perspectives by scientific influencers at the tenured and junior academic and clinical levels. Today, we are pleased to feature Drs. Garnet Anderson and Elizabeth Cespedes Feliciano. Dr. Garnet Anderson is a biostatistician and clinical trialist who focuses on chronic disease prevention and women's health. She's best known for her leadership role in the Women's Health Initiative, a national effort to test methods to reduce the risk of cancer, cardiovascular disease, and fractures in postmenopausal women. Involved from the beginning of the program, she has been instrumental in the design, implementation, analysis, and reporting of the Women's Health Initiative. As principal investigator of the Fred Hutchinson-based Women's Health Initiative Clinical Coordinating Center, she oversees the integration of many different studies within the large program. Of note, she co-leads the Life and Longevity After Cancer, or LILAC study, with Drs. Betty Kahn and Electra Paskett. LILAC is a program to examine cancer and aging among Women's Health Initiative cancer survivors. Dr. Anderson's talk today will describe the available data and resources within the Women's Health Initiative that can be harnessed to support studies of accelerated aging and resilience among older women with cancer. Dr. Elizabeth Cespedes Feliciano's research focuses on body composition and cancer outcomes. Her interests include clinically feasible approaches to measuring body composition in oncology practice, sarcopenia as a biomarker of aging, and understanding the impact of body composition on morbidity, mortality, and treatment outcomes among cancer survivors. More recently, she has begun investigating trajectories of aging in the Women's Health Initiative, including physical function and frailty after cancer diagnosis. Dr. Feliciano will speak about her research on body composition and cancer outcomes, as well as opportunities for understanding trajectories of aging and physical function in aging cancer survivors using data from the Women's Health Initiative. Dr. Feliciano will be presenting first, followed by Dr. Garnet Anderson, and a brief Q&A session. And at this time, I will turn it back over to Jennifer Schaeffer.
Thanks, Jen. And with that, Elizabeth, please feel free to share your screen and start your presentation.
Thank you. All right. Okay. Can you see everything all right?
Yep, it looks great.
Excellent. Well, thank you so much for the opportunity to present at a webinar, named for such a luminary in the field. This is a great series and I've been enjoying participating and now with the opportunity to present, contributing. So I wanted to start by sharing with you how I became interested in body composition as a biomarker of aging and prognostic biomarker in aging cancer patients. And it really came out of my training was as an obesity epidemiologist, and I was studying risk factors for cancer. And it wasn't until my postdoc that I kind of pivoted to study prognostic factors at and after cancer diagnosis in survivors. And when I started to do that, one of my very first studies was, this is unpublished data from an analysis I did at the beginning of my postdoc in colorectal cancer patients at the health system where I work, Kaiser Permanente. And we looked at over 3,000 colorectal cancer patients. And I was surprised to find that body mass index didn't have a consistent role in prognosis across age and cancer site, and even depending on when body mass index was assessed in relation to cancer diagnosis. And so this figure shows on the y-axis the risk of death, and on the x-axis increasing body mass index. And you can see that in the youngest patients in our cohort, those who were younger than 50 years old at the time of their colorectal cancer diagnosis, this line has the kind of expected monotonic increase in risk with increasing BMI. But as soon as you move to the older age groups, the shape changes and it becomes sort of a flattened L shape in which there's an uptick in risk in the lower range of body mass index, even a suggestion of a protective association, and not an uptick again until a body mass index over 35 kilograms per meter squared for body mass index at diagnosis. And so this to me was very curious, and I wanted to understand why. And it sort of prompted my interest in body composition, because body mass index is an excellent proxy for adiposity at the population level and even in younger patients, but it doesn't distinguish muscle from adipose tissue. It's really a measure of body size that's scaling weight to height. And one of the most profound changes that we experience with age is in our body composition. We experience precipitous declines in muscle mass with age, and also a redistribution of our adipose tissue from underneath our skin to around our organs and deposition in other tissues. So here I'm showing you a figure from community dwelling participants without cancer, and the percentage of muscle mass as a proportion of body weight is shown on the y-axis, and then increasing age is shown on the x-axis. And you can see that muscularity declines in both men and women with age, more precipitously in men than in women, but that at every age, there's quite a lot of variation in muscularity or muscle mass as a proportion of body weight. And so this kind of opens the possibility, could body composition be a prognostic biomarker in aging patients, and specifically in cancer? So today I'm going to talk about assessment of body composition, or the assessment methods that I've used, as well as some of the associations we found between body composition and cancer outcomes. And then I'll talk about what I think some of the future directions are for this research in order to make it useful in aging cancer patients. So much of my work, and really the majority of observational research in oncology on body composition, uses clinical CT scans. So CT is collected, computed tomography is standard of care in many cancers, certainly in colorectal, for diagnosis and surveillance. And it's actually a gold standard method to assess body composition. The scans are, of course, not collected for this purpose, but they can be used for it. And so what you're looking at here is a single slice taken from the third lumbar vertebra from the abdomen. And then using a commercial software like Slice-O-Matic, you can parse out the different tissue areas. Red is muscle, the yellow is the visceral adipose tissue around the organs, for example. And most of the research is using these single slice CT scans from the abdomen, partly because the third lumbar vertebra is a very good proxy for whole body volumes of these tissues. And when you do this across a variety of studies, you see that it's prognostic, that low muscle area as measured by CT is associated with an increased risk of death. This is data from a meta-analysis. Actually, the last presenters on this webinar, Dr. Williams and Dr. Muzz, are authors of this meta-analysis, and they found from 38 studies with nearly 8,000 patients that there was a 44% increased risk of death associated with low CT muscle area. One of the reasons that CT is an appealing modality for assessing body composition is not just that it's ubiquitous in cancer, but also that you can get other metrics out of the imaging. not just quantity, but also the quality of the tissue can be measured. And so CT radiodensity is one of the ways that tissue quality is assessed. And here I'm showing you a schematic of how muscle radiodensity is assessed. So you have the zoom in from the area tagged as muscle. This is a zoom in on the postural muscles, and you can see there's different colors there. The different colors correspond to the different Hounsfield unit ranges, so the different radiation attenuation values within that area tagged as muscle. And even within this single muscle group, you can see that there's adipose tissue, so marbling, if you will, that's the blue, the light blue. There's also normal lean muscle, that's the red. And then within the tissue that's actually designated as muscle, there's these lower density areas that are really closer to adipose tissue, but still considered muscle. That's the dark blue and the yellow. And this is considered low muscle radiodensity. And it's indicative of lipid deposition within the muscle cells, so intramyocellular lipid. And that is something that is not only related to strength and physical function, but it's something that occurs with aging and in various pathologies, including cancer, that the radiodensity of muscle declines. So lower muscle radiodensity is also related to overall survival after a diagnosis of cancer. Here I'm showing you an example from that first cohort that I showed at the beginning, colorectal cancer patients at our health system. And what you can see is the risk of death is on the y-axis, and then muscle radiodensity in continuous Hounsfield units is on the x-axis, and there's an increased risk of death at the lower end of the radiodensity range, at the lower end of this spline. So these metrics, these muscle metrics or body composition, in this case, I'm using muscle as an example, it's not just related to long-term survival after a cancer diagnosis. It's also related to shorter-term clinical outcomes that we care about. And this is a paper we had come out recently in JAMS Surgery, in which we saw that low CT muscle area, that's the blue bar, and low radiodensity, that's the green bar, they increase the risk of various outcomes after a surgery after a colon cancer resection. So, for example, patients with low area and low density have a longer hospital stay, higher risk of a hospital stay over 7 days. Also, some indication that there might be an increased risk of surgical complication or readmission, at least for low area, shown in the blue bars. And, well, fortunately, very few patients die within 30 days of their surgery. There is some indication that there might be an increased risk of death after surgery for patients with low muscle area and low density, kind of raising the possibility that maybe these body composition metrics are almost like a embodied medical record or an objective method of assessing the overall state of health of the level of frailty of the patient prior to their surgery. In addition to surgical outcomes, another example of short-term outcomes that are predicted by this by body composition is chemotherapy tolerance. This has been shown in many studies. We've seen it in terms of hematologic toxicity for patients on FOLFOX. But what I'm showing you here is from a review by my colleague Carla Prado, in which she found that the proportion of patients experiencing a dose-limiting toxicity during chemotherapy, that's what's on the y-axis here, that proportion is higher amongst patients who have low lean mass as measured by DEXA or low CT muscle area as measured by CT. And those patients are in green. And the prevalence of dose-limiting toxicity is higher than that shown in the blue bars, which are patients with higher lean mass from DEXA or higher muscle area from CT across a variety of chemotherapy agents shown on the bottom there. Okay, so body composition, or in the case of my examples, muscle characteristics, they're related to short and long-term outcomes in cancer patients. And that sort of makes them a potentially useful biomarker. But I think that there's many areas to explore if we want to make this data useful clinically. And one is we need to improve measurement. Another is we need to understand underlying mechanisms. Is body composition kind of a downstream effect of, for example, the tumor, or it's the cancer treatments one receives, or does body composition impact the course of the cancer itself? Is it both? These are questions we need to answer about mechanism. And then very importantly, I think when and how to intervene to improve outcomes is something we need to understand. So one of the ways to improve measurement that I'm excited about are dilution methods. Because CT, I'm a big fan of. It offers a lot of advantages that I'll talk about next, but you're not going to use CT prospectively to measure response to an intervention or even to monitor the evolution of a patient's muscle mass in the clinic, because in addition to the expense, it's not safe. There's ionizing radiation. So if the patient's not getting CT for some other clinical reason, you're not going to give them CT to assess body composition. So we need safe and effective methods that we can use prospectively. And one of these potential methods is D3 creatine dilution. And so this is a method that we're trying out as an ancillary study to a randomized control trial. And the trial is randomizing patients beginning chemotherapy for colon cancer to resistance training or standard of care. And then we're adding on this measure of muscle mass. And so what D3 creatine dilution does is you take this labeled dose of creatine. The vast majority of creatine, virtually all of it, resides in the contractile muscle fibers. And the conversion of creatine to creatinine occurs at a known rate. And so about 2 to 6 days after you ingest the creatine pill, we can get a single spot fasted urine sample and then calculate quite accurately whole body muscle mass. And part of the reason this is exciting is that in aging patients without cancer, it has very robust associations cross-sectionally with physical function and longitudinally with functional decline and the emergence of disability, and much more so than DEXA lean mass in some studies. And so we don't know how well this works in cancer patients on active treatment, and we're excited to find out. So we launched this study in February, and I think it'll hopefully contribute to improving measurement. The other piece of measurement, pivoting back to CT for a moment, that I wanted to share with you is that there's many groups now that have developed 3D segmentation of computed tomography for body composition assessment. One of those is a group I'm working with that published this paper earlier this year, in which they described their deep learning method for selecting the L3 slice and then segmenting it automatically. And they can now do this across, and many groups can now do this across multiple levels of the CT scan, opening the possibility of looking, for example, at specific organs or at the distribution of muscle and adipose tissue within the abdomen. And we're now using this data in a project on surgical care that's supported by the National Institute of Aging. And a lot of the patients it's a cohort of older adults getting abdominal surgery, and a lot of the patients are undergoing oncology surgery, many of them colorectal cancer resection. And we're hoping to use CT to derive a standardized metric of frailty, an objective and standardized frailty metric that we hope will predict surgical outcomes, such as complications, such as prolonged hospital stay, discharge to a non-home location, or mortality. And so then this data could potentially be used in a variety of ways. It could be used for risk stratification to, if you're not going to change your decision on whether to operate, perhaps you could at least counsel the patient or the family on what to expect after surgery. What will longevity and quality of life look like? Do they need to plan for a skilled nursing facility? Or to risk stratify the patient and provide enhanced pre or rehabilitation. In addition, we'll have the opportunity to explore some of the metrics from the 3D segmentation. And this is an example of what that looks like: the adipose tissue in the abdomen on the top and the muscle on the bottom. So I think that, deep learning methods for 3D segmentation of CT open not only the possibility of asking etiologic questions in large cohorts, but also making this fast and easy to derive from existing clinical data, which could make it useful, at least in prediction. The other area that I mentioned on mechanism is one that I'm very interested in, and we started a project in July, where we are asking the question whether body composition impacts tumor biology. So we're looking at whether the kind of whole body metabolism as represented by the metabolic syndrome and also the patient's body composition measured on CT, does that induce immune and metabolic aging in the tumor microenvironment? If you have low muscle mass and sarcopenia, if you have excess visceral adipose tissue, does that change at all what's happening in your tumor? And so the cartoon on the left is a schematic of a tumor inflammation signature that's supposed to represent kind of a pre-existing immune response to the cancer that is predictive of response to a variety of therapies and improved outcome in some subtypes of breast cancer, but also is thought to be dampened in older patients or in patients who are have an older biological age as measured by their body composition or their metabolic state. So I'm hoping that that by looking at the expression of genes in immune and metabolic pathways in the tumors, that we'll be able to at least open some new hypotheses about why body composition is prognostic in cancer survivors. But this study is really looking at whether body composition impacts the course of the cancer. You could also ask the question in reverse: how does the cancer, and specifically the treatment of the cancer, impact your body composition, impact the sequelae of your body composition? So muscle mass is kind of an underlying cause and fundamental component of the frailty syndrome. And it's possible that cancer treatment induces muscle loss or results in molecular aging, and then induces the emergence of aging phenotypes like sarcopenia or frailty. So this figure here is from a recent publication on adult survivors of childhood cancer. And in this study, they looked at a biomarker of aging, P16 expression in peripheral blood T cells. That's what's on the y-axis here. And this is really a measure of cell senescence. You can think of it as a marker of molecular age. And what they found is that on the x-axis here, you have buckets of intensity of treatment. So sort of a consensus metric of how intense was the treatment that this patient received as a child. And what you can see is that P16 expression is higher in those who had more intense treatment regimens. It's kind of suggesting that here the cancer treatment is inducing molecular aging. And also in the study, they compared frail survivors of childhood cancer to robust survivors of childhood cancer and found that P16 expression was higher in the frail survivors. And frailty is known to be extremely prevalent in adult survivors of childhood cancer. Again, kind of suggesting this link between cancer treatment, molecular aging, and then the emergence of these phenotypes like frailty later down the line. And so there's fascinating literature in adult survivors of childhood cancer, but there's surprisingly little research, at least in terms of long-term follow-up studies, in people who are diagnosed with cancer as adults or older adults. And this is really, I think, somewhere we need to go next with this research, because it will help us understand how and when to intervene. And one of the ways that Dr. Anderson and I have been trying to do this is using data from the Life and Longevity After Cancer LILAC study within the Women's Health Initiative. And here I'm showing you a figure from a recent publication of ours in which we looked at a frailty score kind of aligned with the Fried criteria for frailty. That's on the y-axis. And we fit a linear mixed model, kind of a broken stick model, where we allowed the slope of change in the frailty score over time to be altered at cancer diagnosis. That's time 0. And we looked at how this changed over the many decades of data collection in the Women's Health Initiative. And what we saw is that frailty, as measured by this score, increases over time, but that there's an inflection point at cancer diagnosis, that the slope is higher afterwards, and that this differs actually, intriguingly, by cancer site. And so to me, this study is just the very beginning and opens a lot of questions. So if we know that here, cancer diagnosis seems to be an inflection point at which the rate of increasing frailty accelerates, and this differs by site, why is that? How does this compare? Is this a function of the stage at diagnosis? Is this a function of what kind of treatment you received? For example, is it just patients on anthracyclines who experience greater increases in molecular aging, and then that results downstream in the emergence of the frailty phenotype? How does this compare to age-matched controls who never got cancer? What does their trajectory of frailty scores look like? So there's a lot more work to do here. And one of the only studies in which we can really ask questions like this is the LILAC study. So I'm excited to hear Garnet's take on that next. But before I wrap up, I just want to kind of leave you with a couple take-home points from the areas I've covered. And one is that there's a lot of modalities available to measure body composition. CT is one that I'm a fan of because there's so much digital data just sitting out there that could be used to improve care. And when you use these data, when you assess body composition, you get rich information that's not captured by body mass index that could be used to improve care in aging patients. And the reason I think that is that we've shown, and others have shown, that body composition is prognostic in aging cancer patients, and that it's associated with frailty and functional decline, and that therefore it might be one way that we can develop an objective and standardized metric of frailty in patients, and that would be quite useful, I think, clinically. But before we do that, we really need to understand aging trajectories, because if we know what the natural history of muscle loss or the timing of the emergence of frailty is, tracing patients from before to during to after cancer diagnosis, that's not only going to enable studies where we can ask mechanistic questions about molecular markers, but it's also very importantly going to inform intervention timing. When along the cancer continuum can we make a difference in patients' outcomes? So I think with that, I'm going to turn it over to Dr. Anderson, or back to the moderators.
Thank you, Elizabeth. Garnet, you have screen controls and you can begin your presentation.
Great lead in to what I want to talk about, which is really to describe this resource that we've developed and hope to engender some new collaborations. So I really want to start out with saying a couple words about Arthi. We are coming up to the second anniversary of her passing this week, and many of you were much closer to her than I, but her death was seared in my memory because of my intense relationship with her in the very last few months of her life. My colleagues Betty Kahn and Electra Pasquette had recently introduced her to me and to our group. That we were submitting the renewal of the LILAC Cancer Cohort Infrastructure Grant, and the first submission had encountered modest enthusiasm by the reviewers, and we realized we really wanted to focus on cancer and aging. And Arthi was very generous with her time. She engaged intently with us, learning about this resource and helping us understand what the research gaps were in terms of cancer and aging and how we might contribute to that. So I enjoyed many phone calls and email interactions, and I did get to meet her once in person. And on November 5th, we submitted the renewal grant, and within 24 hours, she was gone. So we are doing our best to carry on the vision that she helped us set, and we are looking for collaborators to help us in that. So basically, many of you know these data better than I do. The number of cancer survivors increasing in the US because of two factors: the aging of our population in general, and the greater survivorship and the success of treatment and early detection. And those are both very positive things. But of course, this has practical impacts on our clinics and our public health system. So it's important that we start to understand what this means for us. There are, of course, very interesting biological questions associated with this. Aging has been long known to be the most common risk factor across all cancers. And we don't really understand whether age is just a convenient measure of cumulative exposure to various factors, or whether indeed aging is driving cancer through one of these mechanisms shown in this grant. But now with the increasing database of cancer survivors, we're also beginning to see how cancer, in particular cancer treatment, may be impacting these same mechanisms and driving aging. So these bidirectional arrows indicate sort of the quagmire of our data, trying to understand how does cancer and aging interact. So, and then in trying to understand what does aging mean, the actual number of days you cross off on the calendar isn't really the key point. The key point for most of us is our ability to function. And so this is a graph that Arthi developed for us. It's similar to ones you've probably seen in other places, where we're looking at function over time. At some point in our lives, we start to lose function, even without cancer. And so there's a trajectory of loss of function that eventually, for some people, would reach a point of becoming disabling, as indicated by the green dotted line. For people who have cancer, they take a hit in their function, but they usually experience some resilience. They rebound, and whether they rebound to the level that they would have had they not had cancer, or not quite as much, is the question. And then more importantly, what happens after that? So if they follow the same trajectory as after the diagnosis as a person without cancer, we would call that a phase shift. So they've experienced a loss, a simple loss, which I think of as a change in the intercept of this decline, but not a change in slope. The accelerated aging hypothesis, of course, is that after their recovery, their decline is much faster than would have happened if they didn't have cancer. So those are some of the questions we'd like to think about. So what are the research gaps in cancer and aging that a large cohort could address? Well, first, we know that there have been very few studies looking at the interactions between cancer and its treatment on long-term outcomes, particularly among those who are older than 75 and among those with comorbidities or other losses. Clearly, the clinical trials of treatments tend to exclude those types of individuals. Older people are really underrepresented in cancer clinical trials. So we need more data to understand how cancer and its treatments interact with these underlying vulnerabilities. There's been a lot of work on geriatric assessments that they need to be incorporated into oncology research. They're needed at the time of diagnosis to help us understand what treatments are appropriate, if we address those earlier, the gaps in understanding, and also to assess these other as endpoints, because things like mental health, physical function, and quality of life may be as important as survival in these patients. And finally, we'd like to be able to incorporate biomarkers of aging into our research. These aging biomarkers, some of which Elizabeth just talked about, can help us identify those at risk for some of these side effects of treatment that might be very debilitating. We also want to look at tumor samples to see if we can determine whether the tumor biology changes in an elderly population. So we're proposing to do this in the context of the Women's Health Initiative. So this is a large program launched by Bernadine Healy as her signature project when she was the director of the NIH. Its main goal was to test in full-scale randomized trials 3 chronic disease prevention strategies, and also to identify risk factors of the major causes of morbidity and mortality in postmenopausal women. The design was focused around the clinical trial component. So it was a partial factorial design testing hormone replacement therapy for the reduction of coronary heart disease risk, calcium vitamin D supplements to prevent hip fractures, and a low-fat diet trial to reduce the risk of breast and colorectal cancer. And then there was an observational, and so there were 68,000 women randomized to 1, 2, or all 3 of those trials. In parallel with almost identical data collection, we had an observational study cohort of almost 94,000 that we're continuing to follow. So in total, we had over 161,000 women, postmenopausal women, generally healthy, who were recruited into this program. These participants were broadly characterized at baseline with extensive data collection that were justified by all these causes of morbidity and mortality that we expect to see in an older population. So I've highlighted some of those in red here that really are important as we think about an aging population: their medical history, the medications and supplements they use, their diet, and other aspects of their lifestyle, mental health, quality of life, activities of daily living. We did a brief clinical exam on all of them. So we have anthropometry. We collected fasting bloods on all of these people at baseline. And we have some more intense measures in subsamples, such as bone densitometry, cognitive assessment, and functional status. So the timeline, these women were recruited between 1993 and 1998. And those in the clinical trial, we actually saw in clinic every year until the end of the trials in 2005. The observational study we saw at baseline and at year 3, and they were followed annually by mail till the end of that protocol in 2005. And then since then, all the women who've consented have been followed annually. So we have up to 25 years of follow-up on these women. The follow-up has been also quite extensive. We've documented all the major health events that have occurred, all the cancer incidents, cardiovascular disease, hip fractures, and mortality. We also have a lot of self-reported events for other health status conditions that are important. We've linked this database to Medicare and to the NDI, so that's expanded and completed the data set where otherwise it would have had some holes. In some cases, we've updated self-reported exposures, and we've certainly focused on some of the patient-reported outcomes, such as quality of life and activities of daily living. And we have additional measures in subsamples, such as anthropometry, blood pressure and pulse, physical function, and densitometry. So who are these women? They were age 50 to 79 at enrollment, that's shown in the green here. As of the last time we took a database snapshot in February, they were between the ages of 72 and 103. There are 63,000 plus who are alive and still in active follow-up, including over 2,700 who were 95 years of age or greater. We worked hard to get a representation from women of color. At enrollment, we had 83% were white and 70% were women of color, including over 14,000 women who were Black and almost 6,500 Latina women. Retention of our women of color over time has not been quite as good, but we're now about 14% of minority participants, but we still have a reasonable contingent of Black women with 4,200 Black and 1,800 Latina women, and we work hard to maintain them as their information is really valuable. Cancer incidence through WHI has been documented on each incident event, and so you can see overall numbers here. We've documented 35,000 new cancer cases and 15,000 cancer deaths since enrollment. The 8 cancers shown here are the cancers we focus on in LILAC. I won't go through all of these numbers, but I can say that the pattern of cancer incidence in the WHI population closely mirrors that we see in the SEER registries. So now I want to go on to what the LILAC is contributing to the WHI resource. So when we initially launched this in 2013, our idea was to supplement the WHI resource with data and specimens that were particularly needed to study cancer survivorship. So that meant to go back and collect cancer treatment and recurrence data and patient-reported outcomes that are specific to cancer and its treatment for the 8 selected cancers I showed earlier. We also decided to supplement the WHI biorepository that has serum and urine with paraffin-embedded tumor tissue from the 6 solid tumors that we designated. And then to use this all to develop a strong research program in cancer survivorship within the WHI. So we published the design and the baseline characteristics of the sample at the end of our first funding cycle. About 48% of the 8 selected cancers were eligible, and we were pleased that for those who were alive, 78% responded to our invitation to be part of LILAC. Most of these cancers were diagnosed in 2001 or later, but the age range at the time of diagnosis ranged from 50 to 90 plus. So we really have a strong data set over those older ages. And this is the status of the resource. In general, we have almost 13, at the end of the first cycle, had almost 13,000 women with 1, 2, or all 3 the basic components, the baseline questionnaire, treatment information, and tissue. So to focus on the treatment, we obtained treatment information either by direct medical review of medical records or by wherever possible, as cost efficiently as possible, we use the Medicare linkage. So we have treatment information on almost 12,000 women. We've also collected tumor samples from around the country. So these women were diagnosed in all 50 states and by thousands of different institutions. So we have collected over 4,000 samples from over 4,000 women that are now available for analyses. So in our renewal with Arthi's help, our vision was to continue to enroll WHI participants who are newly diagnosed with any of these LILAC cancers and to follow up the cohort for cancer outcomes. But more importantly, we're interested in assessing the trajectories of aging and accelerated aging. And we're using the strength of the WHI because we're looking at it in women who have cancer and comparing them to age-matched cancer-free controls. So we can examine the trajectories in women after cancer and look at them in similar data from women, very similar from women who remained free of cancer. And to do this, in part, we're also planning to conduct a home visit among about 2,000 cancer survivors to get specific objective measures. So Elizabeth was talking about how important some of these measures are to really understanding the frailty. We want objective measures on some factors. So by actually seeing these women again, we can have objective measures of physical function. We can obtain post-treatment blood samples that can be used for biomarkers of aging. And we can pair this to age-matched cancer-free control group who will also be receiving a home visit in the context of the WHI. So with Arthi's help, we evaluated the WHI data collection for the geriatric assessment, which we found was so important, and determined that WHI has assessed all the domains that would typically be incorporated in geriatric assessment, but the data are somewhat complicated because they're collected at varying points of time, and some are measured only in a subset of our participants. LILAC has repeated some of these measures during post-diagnosis follow-up, so that helps complete the data set some more, but it's still a rather complex setting. So this suggested at Arthi's recommendation that we use the cumulative deficits approach by Rockwood and colleagues to take best advantage of this existing database for the geriatrics assessment. So I just want to give you a visual picture of what the complexities of using an existing sort of epidemiologic cohort for this kind of question is. So think of this as the WHI data collection protocol. So we collect data annually from these women over time throughout their follow-up period. We collected blood at baseline and again at year 1 in the clinical trial and year 3 in the observational study, and then in a small subset at some other points of time. But of course, the cancer occurrences don't conform to protocol. They don't line up with any of our specific data collections. they happen at the most inconvenient time, both for participants and for us. So how is it that we put that database with this idea of how we're studying aging? So we have to align this around the time of the diagnosis. So for a woman who has a cancer diagnosis early in her WHI enrollment, she'll have some information, she'll contribute some information pre-diagnosis, including a blood draw, but she'll contribute a lot of information about the post-diagnosis experience. For a woman diagnosed much later in her WHI participation, she will contribute a lot of information in the pre-diagnostic trends, but not so much yet, perhaps, on the post-diagnosis trajectories. Importantly, we acknowledge that data collection didn't happen frequently enough around the time of diagnosis for us to really characterize that, that dip in functional status that you might expect right around cancer and the acute periods of treatment. But what we do have is these age-matched cancer-free controls. So for data items where we've collected it frequently, we can compare aging trajectories in women with cancer and those who remain free of cancer in both the pre-diagnosis period and the post-diagnosis period. For the data items that aren't collected as frequently, such as our blood, we can use these matched cases to compare the WHI baseline to the selected additional time points, including post-diagnosis time points that LILAC and the WHI will add to our home visit. So this is an example. We've been playing with the data. These are from our the WHI database that are not our cancer cases or our controls matched to them, but a test set, a separate test set that we're testing these models in. So these are growth mixture models, where we're trying to understand what are the trajectories of aging that you normally see in a population of postmenopausal women. And these analyses, so this is about 12,000 women, show that there are 4 what's called latent classes, profiles of physical function that occur over the 20 years. You can see there are 2 groups that start out with relatively high, most of that physical function over the 20 years of follow-up that we have. The next group down starts out pretty high, but about halfway through this period starts to suffer some significant decline in physical function. And then the 4th group starts at has already experienced a modest level of impairment or loss of function, and they continue on a rather smooth trajectory of loss over the next 20 years. So as you might guess, we looked at predictors of the membership in these latent classes, and there are no surprises here. Age, BMI, self-reported general health, physical activity, and falls in the last year were all really important predictors of which of these classes a woman would fall in. So now what we're going to do is, after we're comfortable with this modeling, take them into the data set of LILAC cancers and their age-matched controls to see how these trajectories look in the women with cancer and the very comparable women without. We're very much looking forward to our home visit protocol, which we anticipate doing in early 2022, starting that, assuming that the pandemic has waned. We will repeat elements of our baseline data collection, including a brief physical exam. We'll do the short physical performance battery for objective assessments of frailty, and get a new fasting blood collection. Again, we hope to do about 2,000 LILAC participants, and WHI will be doing actually around 4,400, we anticipate, additional women. So the follow-up will be the women who had such a visit in 2012 and 2013. There were about 8,000 who had that. The age distribution is here. We oversampled our Black women and Hispanic women, so we'll have a strong ability to look at those subgroups. As of This year, a large number, we have 5,500 of those are still alive and in active follow-up, including a small number who are over 100 years of age. So we're hoping to have a good representation that these women will agree to a repeat visit so we can see what's happened in the decade since that last visit. About 1,000 of these women had a cancer, not necessarily a lilac cancer, but a cancer prior to their home visit in 2012, 2013. So we'll have a chance to see some of those in the second visit. There are another 700 who had a cancer diagnosis after that. And so a second home visit will allow a blood collection and other measures to be taken after that diagnosis, which will help in those trajectory analyses. We're particularly interested in that blood collection. Elizabeth defined a few biomarkers, the D3 and the P16, that would be interested in testing for effects of cancer on aging. This is another one that Betty Kahn is particularly interested in, the epigenetic clock. There are many others who will be interested in using these specimens to help us understand the biology of cancer and aging. So I just want to come back to Arthi. One of her favorite sayings was, if you want to go fast, go alone. If you want to go far, go together. We've used this opportunity to help develop a partnership with the Cancer and Aging Research Group, particularly William Dale and Supriya Mohili. We are the Cancer and Aging Research Group have a number of data sets for their group to use, and we want to make LILAC one of those to make sure it's used in to the extent that it can be, that it can contribute as much as possible. And so we're looking at ways that we can jointly support pilot studies and offer the LILAC resource to people with expertise beyond our own. So if you are one of those people interested in collaborating, I do encourage you to reach out to one of us. I've given our contact information here. I hope you will take the chance to come up with ideas and propose them to us. I would like to highlight our website, where you can get a lot of information about the WHI resource, which is very rich and very open to collaborations from people all over. So with that, I'd like to thank you, and I will stop my presentation, and we'll be happy to have questions.
Thank you, Dr. Anderson and Dr. Feliciano, for your presentations. They were both very informative. We do have a couple minutes for questions. The first question I will ask is, and it's sort of, is at an intersection of both of your presentations. Are there any body composition data in WHI, or can that type of data be accessed? And I'll throw it out to either of you.
Okay, so body composition data. So we measured BMI on all the women at baseline and when we had clinic visits or in-person visits. So height, weight, waist, and hip circumference. In a subset of about 11,000, they had bone densitometry. So that is, I'll let Elizabeth say a little bit more about the quality of that. That is giving us some information on body composition. But Elizabeth, maybe you should say a little bit more about the quality of that data.
Well, I think that the availability of DEXA in the WHI is a huge advantage. And I've myself worked with the combination of even just BMI plus waist circumference, which gives you some additional information on a very large number of women at least 2 visits, 3 years apart. I think what, maybe Garnet, you know the answer to this, I don't right now, but I'm not sure how many of the women who received DEXA eventually developed cancer, or when the DEXA measurements are in relationship to cancer, because I know some participants actually have multiple measurements of DEXA over time. A general comment about DEXA is that there is some evidence that in aging patients, lean mass might not be the physiologic variable that we're most interested in, just because it's not just muscle. We treat it sometimes like it is. We calculate appendicular lean mass from DEXA, partly to exclude things like organs that might otherwise be in that lean mass measure. But still, at least in some cohorts, Mr. Oz is an example, DEXA lean mass isn't always strongly associated with functional outcomes. But I think that it's something that could be incredibly useful to assess body composition in the WHI, and that there are existing studies that are deriving visceral adipose tissue, for example, from the existing DEXA scans, which were analyzed on an older software and didn't originally have it. So I think it'll be a great resource.
Great, thanks. So another question, a question about accessing the WHI resource. Are the collaborators open to studies that are intervention studies or studies that involve recontact of participants? Or do the studies proposed need to simply be secondary data analyses and studies of that nature? So I'll ask Garnet to address that one.
Yeah, in fact, we're actually very excited about interventional studies. And in fact, it may be that at some point our continued funding has been conditional on doing that. We want to produce the highest quality evidence as possible, and we recognize that interventional studies are those. So we actually have two ongoing intervention trials in the WHI cohort. One is testing a physical activity intervention for an aging population. That's called WHI. It's organized by Marcia Stefanik from Stanford, Andrea LaCroix from UCSD, and Charles Cooperberg from Fred Hutch. So that trial is ongoing. There's another one testing some dietary supplements, multivitamins and cocoflavanol supplements in a subset of WHI women. That's called Cosmos. And that's led by Joanne Manson and Howie Sesso from Brigham and Women's and myself. So we are very interested in additional intervention studies or studies that would involve additional contact of these participants. What we are careful about is participant burden. So we look at that and try to understand whether these women, how much they're already being asked to do, and try to make sure that we're not overburdening them.
And we've got a couple questions about the age match controls, and could you elaborate a little on how the age matching is being completed? Are you matching only on age, or are you matching on other factors as well?
We're matching on a few other factors, and we can make multiple control groups. We have a wealth of controls, but to start with, it was age at diagnosis and time in the WHI, so they would have similar amounts of data collected prior to that, as well as we can match on comorbidities at baseline or at the time of diagnosis. And let's see, also where in the region of the country they came from, which original study component they came in. That's pretty important, matching on clinical trial versus observational study, because the clinical trial women had annual in-person visits, so their data collection is richer, and we would want to make those similar. So we have a sort of a template for how to create a control group, and we plan to use one of the control group that we have made for most of our analyses. But if someone has a particular need to select a control group differently, that's also possible.
Okay, I think it's nearing the top of the hour, so we will end our Q&A session, and thank everybody for attending the webinar today, and especially thank our two speakers who informed us of the work they're doing. And for everyone, we're putting together our list of speakers for 2021. If you have any suggestions, please email the team, but stay tuned for our emails announcing the dates and times for our webinars, and we hope to send that out in the coming months. Thank you all, and have a nice rest of the year.
Thanks, Lisa.
Thank you.